So today we are going to discuss about the complex diseases. Some diseases are basically common diseases, normal diseases, which are usually caused because of the genetic effect of. But there are certain diseases which are not caused just because of a single gene. They are caused because of the multiple factors. For example, if we see these could be the multiple factors, including the genetic factors, environmental factors, as well as the lifestyle factors. So, any disease that is caused because of the combination of multiple genetic and environmental and lifestyle factors, that disease is known as a complex disease. For example, if we talk about the cystic fibrosis, this is basically caused because of the single gene, and that is the CFTR gene. This particular disease is caused because of the CFTR gene, cystic fibrosis transmembrane receptor. If there is any mutation, basically F408 mutation, then this leads to the cystic fibrosis disease in the lungs. So this disease is simple to study because this is caused because of a single gene. But there are many diseases which are caused because of the multiple genes, which could be because of the environmental factor, which could be because of the lifestyle factors, what you are eating. So, if the disease that is affected by many genes means it is polygenic in nature, and then it is difficult to study these kind of diseases, and these diseases are known as the complex diseases. So, what is the features of complex diseases? Number one, it is multifactorial. It could be caused because of the multiple genes, means it could be polygenic. Number two, it could be because of the environmental factors. It could be because of the lifestyle factors. This is a very important point. Polygenic inheritance. Many genes contribute small effect that together influence the disease risk. So every gene is having a small amount of effect on the disease. Gene environmental interaction like diet, smoking, infections can modulate the genetic risk. And variable expression, it means some people will show more number of symptoms, some people will show less number of symptoms. So expressibility is different. Okay, for example, if you talk about any disease, let's say the polydactyly, where multiple fingers are there. So some people show only 6 fingers, some show 7, some shows 10. So expressibility is different in that case. So these kind of diseases are known as the complex diseases. So what are the examples of complex diseases? The very good example is cancer. Because cancer is not caused because of a single gene. There are thousands of genes which are causing cancer, and that is why it is very difficult to study this particular disease. Similar to cancer, there are neurodegenerative disorders, psychiatric condition that specifically if we talk about schizophrenia, depression, that are more sort of environmental uh, and that depends on the environmental conditions sometimes, and also the lifestyle factors, how much stress you have. So these diseases, it is very difficult in case of the complex diseases to separate the genetic effect. If you want to separate the genetic effect from the environmental effect, it is difficult. Okay, so what we can say, difficult to separate genetic effect from the environmental effect in this case. Now, as it is mentioned here, sometimes diseases run in family, just not because of the genes, but because of the family members. They share the same environment. For example, children learn the same language as their parents, not because of genetics, but because of the environment they grow up in. So some have a very good sharp IQ, some have a very less IQ, and sometimes they develop the depressions and other kind of diseases. This shared environment can affect their behavior traits like IQ, mental health issues such as schizophrenia, which can be influenced by how someone is raised. Even physical traits of birth defects might be due to shared habits like diet or use of certain traditional medicines. There's a very good example if you see about the Kuru. If you see this family, Kuru family, this is the fourth pupil in the New Guinea, it's a tribe, and almost 80% of the people are developing a disease known as Kuru. And why it is developing, it is not because even from the birth, since the birth they are developing it. And the reason is not genetic. The reason is their tradition. They are eating the brain of the diseased relatives. That is their ritual, and they are following it very strictly. And that is the reason they are developing the Kuru disease. So what is basically the Kuru disease? So it is very difficult to, say this is genetic and this is environment. So it's difficult to separate. Sadly, this is not always clearly mentioned in medical papers. This is the example. You can see that this is the Kuru disease in this child. You can see there is no coordination of the hand. They have a headache. They have problem in swelling. Sometimes they laugh like anything that they can't have any control on their laughing, and pretty soon they die off. So this is the condition which is not genetic. This is the condition because they are eating the brain of the diseased individual, and that is their ritual. Their parents are following, so they are also following. So how you can study the complex diseases? Because you have to study multiple genes. You have to see what is the environmental effect. You have to see what are the lifestyle factors. So whenever the researchers want to study complex diseases, they usually do it in the families, twins, or adopted children. But it's very tricky to separate the effects of genes from the shared family environment. If we talk about the twins, so there is monozygotic twins. So they share 100% gene similarity. Okay. Then there are dizygotic twins. They are sharing 50% gene similarity. Okay. So because they are sharing the genes, and most of the time they are grew up in the same environment and their lifestyle factors are also same. So sometimes it is very uh, easy to study the complex diseases. For example, there is one term which is concordant. What is concordant means? Both the twins are affected. Both the twins are affected. Now, if you see about discordant, discordant means only one twin is affected. So whenever concordance in the monozygotic twins is more than the dizygotic twins, you can say this could be genetic. Similarly, if the monozygotic twins concordance, if it is more than dizygotic twins but less than 100%, then you can say this might be because of the environment. Because if this concordance is, if one twin is affected, another twin is also going to be affected. And then if concordance should be 100% if only the genes are the region, but if genes are not the only region, then it is going to be less than 100%. And then you can say it could be because of the environment. So that's how people study whether the environment is having any effect or not. I hope you understand. When the monozygotic twins are concordant, means if one twin is having diseases, other is also going to be having diseases. Then in that case, the concordance should be 100%. But if it is like more than dizygotic twins, but less than 100%, then you can say the environment is also playing a role in this concordance. So that is the reason why people are studying complex diseases in the uh, twins. That's the reason. So risk ratio is the relatively risk factor. If suppose one member of your family is having a disease, then the chances of risk ratio is going to be more. If two members are having the disease, then chances are even more. If you are closely related to the person who is having disease, then the chances are more. If you are distant relative, then the chances of the risk is going to be less. For example, if your father is having a disease, then chances of you having a disease is more comparative to your grandchildren. Okay, so like that. So whenever the risk ratio is 1, it is no extra risk. Whenever it is more than 1, it is high risk. Whenever it is less than 1, it is protective. So let me write it down here. So whenever it is 1, as I mentioned here, no risk. Whenever it is more than 1, it means high risk. And whenever it is less than 1, it is protective. Okay. Mm. So now, for example, if one parent has schizophrenia, then the child risk is 7 times higher. But if both parents are affected, the risk is much more, then the risk is even higher, 58 times more. But for more distant relatives like cousins, the risk is much lower and closer to normal. This shows that the closer the family relation, how close you are with the person who is having a disease, the chances are going to be very high. The higher the genetic risk. So this is about the twins. Monozygotic twins have the same genes, while the non-identical twins, dizygotic twins, share only about half genes. So if the trait is genetic, monozygotic twins should have it more often than dizygotic twins. Okay, but if it is less than 100%, it means this is not because of the just gene. It could be because of the environmental factors as well. You need to find the concordance. If one twin is having a disease, other twin is also having a disease, same disease, if it is 100% genetic. Okay. But if in case of the monozygotic twins, but if in case of the monozygotic twins, if the concordance is less than 100% than the dizygotic twins, it means the disease is not just because of the genes, it is because of the environmental factors as well. So So concordance, how you can, there is two kind of concordance. And what is the concordance? What is the meaning of concordance? You just want to identify if one twin is affected, what is the probability of the other twin to be affected. If suppose I have my twin sister, I am affected, I just want to know what is the chance that my sister is going to be affected. And that is why we calculate it. So one is the pairwise concordance. So pairwise concordance means if suppose 8 of the 24 twin pairs are concordant, then you can just do the pairwise concordance. You can calculate it. So one is the pairwise concordance. So it is like 8 pairs are concordant out of 24. So 1 by 3. One is the probandwise concordance, which is 2C divided by 2C plus D. C means concordant pairs. For example, here the concordant pairs is 8. And because concordant pairs is 8 out of 24, so discordant pairs will be 24 minus 8, which is 16. So like that. So you can say 0.5, 50%. 50% is the chance that the other twin is going to be affected. So what is basically proband? Proband is the twin which is giving you the indication that the other twin might also be affected. When it is more relevant in comparison to the pairwise concordance. Because in the pairwise you are everything calculating in the form of pairs. If one pair is affected, other is also going to be affected. But in the proband wise, we are not studying pair. We are saying if one twin is affected, what is the chance that the other twin is also going to be affected? So this is the pairwise concordance. So proband wise concordance are thought to be more comparable with other measures of family clustering. Studies show that monozygotic twins with schizophrenia are more likely to both be affected compared to dizygotic twins, and this suggests a genetic risk. Okay? Whenever the concordance in the monozygotic twins is more than dizygotic twins, it means it is because of the genes. Otherwise, it should be similar because monozygotic or dizygotic, they are sharing the same environment. If it is because of the environment, it should be same. But because the concordance is high in monozygotic twins, you can say it is genetic in nature. However, twins studies have also some limitations. For example, monozygotic twins share the same sex, dizygotic twins may not be. Monozygotic twins look similar, same, treated more similarly. They share they may share more of their environment. This means higher similarity in the monozygotic twins might be due to the shared environment, not just because of the genes. And that is the reason why people are using conducting twin studies to find out whether the disease is because of the gene or because of the genetic factor. If it is because of the genetic factor, if it is because of the environment, both the concordance in the monozygotic and the dizygotic twins is going to be same because both the twins are sharing the same environment. That's the assumption most of the time. Another study is adoption study. So what you are basically doing it, two ways of doing the adoption study. Find the adopted pupil with a disease, the person who is having a disease, and find its family and see if the disease is there. If the disease is there, it means it is genetic. If it is not, it might be because of the environment. Or second option could be find the parent with the disease and see the child is having a disease. If the child is having a disease and parent is also having a disease, then it means it could be genetic. But if not, then environment is playing the role. So adoption studies are also very useful in studying the complex diseases. A famous study on schizophrenia showed that the disease was more linked to biological parents than adopted ones. It means it is genetic in nature. The main problems of adoption studies is very difficult to get all information of the biological families, and sometimes the adoption agencies place children in homes similar to their birth families, so their environment is also same. So even with these issues, adoption studies are still the most reliable way to test how much genes influence the trend, especially for mental health conditions like the schizophrenia, depression, like that. Another way to study the complex disease is the segregation analysis. In this, what you are going to do, you are going to basically find it out whether the disease is autosomal dominant. If it is autosomal dominant, you will study the pedigree chart and you will find it out if it is dominant or not. Then autosomal recessive, X-linked, you have to find it out by studying the pedigree chart. If you see, let's say, beta thalassemia, and if the person has, let's say, is heterozygote, mother and father both, then what are the chances? This is going to be the chances of developing the disease, only 25%. Because it's a recessive disease. Both the alleles should be recessive. Only 25% out of four, only one will develop the disease. So accordingly, you can identify whether the disease, how the disease is progressing in the family, and you can find it out if it is autosomal dominant or autosomal recessive, or if it is X-linked. If you find it out these things and you came to know the exact pattern that is expected in the Mendelian inheritance, you can say it is because of the single gene. And then you can do the linkage analysis. You can find the marker gene near to the gene, and then you can see how it is progressing in the family. But if this is not following the Mendelian pattern, then in that case you have to conduct the genome-wide association study where you find lots of SNPs and see how it is progressing in the family. If it is progressing in the family like the way it should be followed, then you can say it is genetic. If it is not, that means the environment is playing the role. So what are the limit of so what you will do in the segregation analysis? Number one thing, you will find it out whether the disease is autosomal dominant, recessive, X-linked, and if it is following the Mendelian pattern, you can say it is because of the single gene. Then it is simple to identify. But if it is not following the Mendelian pattern, because of the, there could be many regions. For example, the regions if we talk about, it could be incomplete penetrance, could be the region that it is not following the Mendelian rule in the autosomal dominant condition. Incomplete penetrance. And a very good example of incomplete penetrance is polydactyly. Many fingers in the uh, hand or leg. Then it could be expressivity. It could be the modifier genes which modify the expression of a main gene. It could be environmental factors. So these are the things. So whenever you are writing about the segregation analysis, you have to write it out. First of all, we will identify it is autosomal dominant, autosomal recessive, X-linked. Then we will find out the pattern. Pattern is Mendelian or not. If it is Mendelian, then it is simple disease. If it is not, it could be because of these many reasons. If it is not, it could be because of one is environmental factor, incomplete penetrance, expressivity, modifier genes. It could be because of that. And if it is like that, then you have to conduct a GWAS, genome wide association studies, to find the actual genes rather than just modeling them. So it can work with the information it's giving. If important factors are left out, the results can be misleading, especially for behavior or traits affected by the upbringing. Today, segregation analysis is used less often because sequencing is available right now. Many diseases have already been studied, and because they are so genetically complex and varied, researchers now prefer more direct methods like the linkage analysis and genome-wide association studies to find the actual genes rather than just modeling them. So how you can do the segregation analysis? One is the parametric linkage analysis. Here you are a mathematician, a creating or a statistician are creating a model. And how they are creating the model? When they know disease, how the disease is inherited, dominant, recessive, X-linked, what is the pattern? How common the gene is in the population? How likely a person with the gene will actually show symptoms, means the penetrance. Is it easy to know all these things? It's very difficult to know. So that is the reason this method is not considered a powerful method, though it is very powerful, but it is not easy to operate. If a good model is available, parametric linkage is a powerful tool. It can scan the entire genome to find the location of the disease causing gene, but you should have all the information with you. Then they will create the model. This model is the standard LOD score analysis method in the parametric linkage analysis. So, what is the method of segregation analysis? Parametric linkage analysis. Or this you can do when all these things are available to you. For Mendelian diseases, which is caused because of a single gene, creating such a model is very straightforward. But for complex diseases, it's much harder. It's not simple because you don't know how much is the penetrance. You don't know how common the gene is in the population because this is not just one gene. There are many genes which are causing complex diseases. And how will you know one gene is common, one gene is not common? Something like that. So even if it can estimate overall genetic risk using segregation analysis, we still cannot accurately guess the specific details needed for the parametric linkage analysis. That is the reason this model doesn't work well for the studying complex diseases. So this model is good for studying the simple diseases, Mendelian diseases, which are caused because of a single gene. They are following the same pattern. But for the complex diseases, it is not simple to follow. Now coming to the another method, which is a non-parametric linkage analysis. It's a model-free approach. Usually, scientists use it because they don't need to study, they don't need to assume anything. They just create a model. And how they create a model? It simply looks for the chromosomal regions shared more often than expected by chance and affected individuals. Let's say this is your mother, this is your mother, and it is having the A and AL. And this is your father. This is also having the A and AL. So if they will cross, if you do this, so you will get 25% AA, 25% AA, small and smaller, 50% will contain one from father and one from mother. So whenever this ratio is not there, suppose these two parents have two siblings, both the siblings, if uh, sharing the same allele, then the chances are going to be, suppose if it is like both the siblings have AA, or all the four siblings have AA, it means 100% they are developing this AA. One is getting one allele they are getting from mother, and one allele they are getting from father. But it should be 25%. If it is more than this, then you can say that this particular locus of chromosome is linked to the disease causing gene. So let's say this is the chromosome, let's say this is the chromosomal number 7. You find it out and you can see that this chromosomal region is shared more than 25% in case of both of these siblings. Then you can say this could be because of the gene disease susceptibility. This is because of the presence of the disease susceptibility gene near to this in this chromosome or in this particular region. So this is known as the identical by descent method. Usually, this is the pattern they should follow. So if these parents have four children and all the four children have capital A, it means 100% they are having capital A, capital A. It means this particular region may be linked to some certain disease causing gene, and that they need to find it out. This is known as the identical by descent, alleles inherited from the same ancestor. Identical by state, alleles look the same, but we cannot say they are coming from the same ancestor. Sometimes they look same, but it could be because of some other reasons. Then these are the identical by state. IBD is more informative but needs more family data. IBD is easier to use but maybe less reliable. IBS because especially with the common alleles. So this is, as I said, siblings normally share chromosomes like this, 25%, 50%, and 25%. But if both siblings have the genetic disease, they are more likely to share the segment carrying the disease gene. If the disease is dominant, affected siblings tend to share one fragment, the one with the mutation. If it is recessive, they will share two segments. Like for example, I see both the beta thalassemia, this is a recessive, so both should be recessive. But polydactyly, which is dominant, so one allele is enough to cause the disease. That's what they are saying. Even for the complex traits, affected sibling pairs share the disease-linked gene region more often than chance. So if suppose this region is linked to the disease, let's say capital smaller recessive region, they will share it more in case of the complex diseases because this region is simply located near to the disease causing gene. And by this you can study the complex disease, complex traits. So whenever the complex traits are studied, they are studied in the near Mendelian families. Near Mendelian families are families which are not following the exact Mendelian pattern, but they are following a sort of Mendelian inheritance. For example, if we talk about the uh, BRCA mutation, so in case of the breast cancer, this gene is mutated, but all the carriers will not develop the disease. Okay, so these kind of families are known as the near Mendelian family. And again, this could be because of the incomplete penetrance, expressivity, environmental factors, lifestyle factors. Even though suppose this is autosomal dominant diseases, let's say if this is the autosomal dominant parent, then their kids should have this disease. Okay, but if at least 50% should have this disease. But if it is not the case, it could be because of many reasons, like incomplete penetrance, like expressivity or environmental factor, modifier genes. So then these are not following the Mendelian patterns, and therefore they are known as the near Mendelian families, and that is why these families are more important in studying the complex diseases. So if many affected siblings sharing the same genetic segment more often than expected, that's a red flag. There may be the disease gene hiding there. Software like MapMaker, Gene Hunter help in finding these patterns. An NPL score is a non-parametric LOD score. If the LOD score is more than 3.6, there is chances of linkage. If LOD is more than 4 in case of the IBS sharing, then significant evidence of the linkage. So NPL score measures how likely it is that the observed sharing is not due to chance, but because of the gene disease-causing gene located near to this particular locus. So you calculate the LOD score, and that's how you can find it out uh, that there is a linkage between this particular segment, which is sharing more than expected, basically, and located near to the disease-causing gene. Okay, if you want to study more on the LOD score, you can go ahead and watch my lecture, linkage disequilibrium. I have explained it there. So with this note, I am closing this lecture. I hope you understand what are the complex diseases. The things which you need to understand is this: there are basically they are basically done in the twins and the adoption adopted children. Then you can use the mathematical model, which is the segregation analysis. But in this case, you have to find it out. You have to study the pedigree, find it out whether the disease is autosomal dominant, recessive, and linked, whether the pattern is Mendelian or not. If it is not, it is because of these three factors. And how you can do that basically? You can do it by the parametric linkage analysis, but because all the information you want to create this model is very difficult, so this is not a good tool for studying the complex diseases. However, in case of the non-parametric linkage analysis, you are not assuming anything, you are just finding out what is the region. If suppose both the siblings have disease, what is the region that is sharing more than expected? First you find it out that thing, then you find it out, then you assume that it could be because of the gene which is causing the disease. So this is the non-parametric linkage analysis. Uh, and usually we do it in the near Mendelian families, because near Mendelian families, uh, whatever is followed in the Mendelian families, it is not completely followed in the near Mendelian families. For example, if this is a dominant disease, it should be there in the 50% of the children, but if it is not, then it might be because of so and so regions, and this is a near Mendelian family. Even though if it suppose if it is followed in 25% of the children, it is followed. Okay, so that is why it is considered a near Mendelian family. Then you calculate the NPL score, and based on that you can find it out whatever the segment. If suppose two siblings have the disease and sharing the one segment more often, means not, let's say they should share by 25%, but they are sharing like 50%, then you suspect it could be because of the gene located near to this particular locus, and this gene could be the disease susceptibility gene. So that's all for my today's lecture. Thank you so much.